sure this is actually recording. Yeah, it is. Okay, we're going to spend a brief overview on a few of the methods used in cell molecular biology and, of course, development. We've got about 30 pages of material here on our first chapter of our lecture note manual. Even though I'm not going to have time to cover all this stuff in class, you are going to be responsible for that. I'll usually throw a question in there on the a question or two in there on the test, so please be forewarned about that. So read that chapter, and of course take notes for what I'm doing here. I'm going to add one or two things I haven't been able to add there yet, but you are going to be responsible for that material, even though I'm just going to gloss over it here and try to get the whole thing done today. So, first of all, does anybody have any questions so far? Any questions? Okay, well, let's take a look. Different kinds of methods of looking at development. Of course, the first ones were the classical ones using a microscope. Typical microscopes like the ones we have in the labs, you shine a beam of light through a very thin salami slice or section of cells, tissues, an embryo, what have you. Various kinds of organic molecules, biological stains, are going to selectively bind and stain certain cellular components. For instance, you have lipid-specific stains, very hydrophobic molecules, they stain the cell membranes. You have stains that just stain just about any kind of protein around. You've got stains that are going to stain DNA and RNA both. You have stains that are DNA-specific, RNA-specific, polysaccharide stains like the Gram stain they use in microbiology and so on. And we saw some of that when we were looking at some of those slide sections on Tuesday. Notice, for instance, when you looked at the germinal vesicle of frog egg, you see those little purple-pink dots that were the nucleoli. That's because they used an RNA-specific stain, and the nucleoli are especially loaded up with RNA, so they stain very nicely. So those are kind of examples. When you look at living organisms or unstained things, you know, cells are pretty transparent. You really can't see too much. The one problem is most stains are very toxic to organisms, so they only work on preserved or fixed tissue rather than living tissue. There are some stains that can be used in living embryos that aren't toxic. They're called vital stains, but people sometimes use that. But there's a number of other tricks we can do to localize things specifically in cells or embryos. We can localize proteins, or rather specific proteins, by a technique known as immunofluorescence. Now the term immuno refers to immune system because what we're going to be doing is generating specific antibodies that will bind specifically to a target protein. And the way you do this is you take a protein, you have to purify it. That's an extensive problem. You purify the protein. Now you take this protein, inject into something like a mouse or a rabbit, some kind of test animal. That protein, even though the mouse or rabbit may well have the same type of protein, is going to be different. It's going to be somewhat, sometimes extremely different from the rabbit version or the mouse version. So the animal's immune system recognizes that injected protein as a foreign protein and mounts an immune response to that, producing large amounts of antibodies that will stick to that protein. And of course, that's how you end up. That's how immunizations work and things like that. So now we get. The animal generates antibodies that are going to bind to that particular target protein. And now you purify those antibodies. Relatively easy to do. And finally, You can add them to a section or a slice or a whole embryo or a section or slice of an embryo or what have you. You can add these to your sample. Let me scoot over here. Okay, you add your antibodies that you've obtained from whatever, the mouse, the rabbit, what have you. They stick to wherever those proteins are located in the cell or the embryo. Now the next thing you do is add a second set of antibodies. You buy these commercially. Let's suppose that our original antibodies, we injected a protein into a mouse, so now we have a bunch of mouse antibodies that stick to our protein of interest. We get a second set of antibodies. They're labeled with a fluorescent dye, so they'll glow in the dark at you. And those antibodies are going to stick to the original mouse antibodies. So we have something like this. Here is a protein. And here's the first antibodies. Antibodies are kind of shaped like Y-shaped molecules. Okay, now they're sticking to that protein. Our second set of antibodies stick to the first set of antibodies. Like I said, and they carry the fluorescent dye, or you can use radioactive isotopes or stuff. People use dyes most of the time. Okay, so now we have a second set of antibodies, and several copies of them are going to stick to each of the original antibodies. So I'll put a star there to tell me that these are labeled. Okay, now you look at it under what's called a fluorescence microscope. A fluorescence microscope is like a regular light microscope with one difference. Instead of illuminating a specimen with regular visible light, it uses ultraviolet light, which we can't see. But the ultraviolet light causes those labeled dyes, those dyes on the second antibodies, to glow in the dark. They may glow at you green, red, blue, there's a bunch of different kinds. So that allows you, when you look at a cell or tissue, say, hey, what cells in this embryo have this particular protein? And maybe how much? So, end result, here's an embryo of some kind. And maybe you find a whole bunch of glow in the dark in one end of the embryo, perhaps the head end of the future embryo, and very little or none in the tail end. That tells you where that protein is located. And you can see the results of gene expression that way. You can see that certain cells may show the presence of a particular protein while other cells do not. Of course, if you have something against one of those so-called housekeeping proteins, you're going to find it all over the place. But when you start looking at specific proteins, you can find them localized to specific parts of the embryo. And if you go in higher magnification and look at a cell, here's an individual cell here, you may find a lot of glowing, say, just beneath the cell membrane or around the cell membrane, suggesting that that's a membrane protein or something associated with the membrane. Or you may find a lot of glow in the dark in the nucleus, telling you that that's probably a nuclear protein. So this kind of technique allows you to localize things in cells and in embryos. So it's a very powerful technique. In fact, the textbook has a number of pictures of embryos or sometimes of cells using this type of method. either fluorescently labeled antibodies or sometimes antibodies that have an enzyme attached to them that leads a colored precipitate or whatever. There's a bunch of different variants of it. But the key idea is using specific antibodies to target and localize a specific protein. That's what we call immunofluorescence. So that's a very important technique. There's a version of it called a confocal microscope, which is described in our little manual here, and that allows you to get even sharper images than you can with a regular microscope. Downside is those things cost about $100,000. But the upside is you get better images, and there's details of that in our lab manual. So that's a powerful technique. Another thing we can do to localize in cells or embryos specific nucleic acid sequences. Okay, it's called in situ hybridization. We'll look at hybridization a little bit later, but quick and dirty. The idea behind this technique is to localize a specific nucleic acid sequence in a cell or an embryo or piece of tissue. That can be either DNA or RNA, depending on what you're trying to look at. In this, the quick and dirty version, in this what you do is you have let's suppose we've got a DNA sequence, a gene here. Let's suppose we want to localize a specific gene. Well, there's techniques that allow you, in fact, actually, this is usually done, you pay some company to do this stuff for you. You give them a DNA sequence or a part of it that you want, and they'll produce in their facility a little short piece of DNA. Now this piece of DNA here, which we often call a probe, and I'm just drawing one strand of it, it's labeled with something. It can be labeled with radioactive isotopes, fluorescent dyes, or any of a number of other things. But that way allows you to detect it. Okay. What it is, it's got a complementary or opposite sequence of one strand of this DNA. Let's suppose this represents a gene here. Okay, so if we were to pull these strands of DNA apart, we'll do that. Let's pull one strand off here. This probe could stick to this other strand by hydrogen bonding. And carrying some kind of label, you could tell and detect the presence of that. And even how much is there. So what we do with this technique here, we take our cell or tissue, now this has to be done with stuff that is already dead. But you take cells or tissues, you expose them to chemicals, something called Forma is widely used. Chemicals that break the hydrogen bonds between two strands of DNA separating. So now we separate the DNA strands inside our tissue or cell. Now we can add the probe, and once again that probe is complementary or opposite sequence to whatever your gene or DNA sequence you're looking for. Okay, so you add that probe. Now, once you remove the chemicals that separate the strands, you just wash them out basically. Okay, when you remove those chemicals, of course the original two strands of DNA could meet each other and stick together again. But there's so many copies of that probe around, it's much more likely that that probe will stick to the strand of DNA first, long before its sister strand happens to come around. So most of the time, the probe's going to stick to your DNA strand. I'll put it right here. There it is. And it's going to glow in the dark at you, or expose film if it's radioactive, whatever the case is. And what this allows you to do is, once again, localize, detect the presence of a DNA or RNA sequence. You can do the same thing with RNA. It will localize it in an organism, in an embryo, in a cell, or in a tissue. And here again, our textbook has some neat picture of that. For instance, using this in immunofluorescence, you could look at an embryo. Here's an embryo. Okay. You could ask yourself, with a specific protein, where is that protein located in this embryo? And if you look at that protein, let's suppose we find a lot of it in the tail, but not much elsewhere. It tells you that that protein is being specifically produced at the tail end of this early embryo. Now, using this in situ hybridization technique to localize nucleic acids, you could ask yourself, where is the messenger RNAs that code for that protein located? Well, you might find, now do the reverse cross-action, you may find that the messenger RNAs are located about the same place that the proteins are, telling you that those messenger RNAs are specifically being produced in the cells in the tail part of this embryo, and the proteins are being made, translated from those messenger RNAs. But sometimes you get surprises. Sometimes you might find the messenger RNAs all over the place. And what that would tell you is that gene is transcribed in the messenger RNA all over, but it's only translated into protein in one part of the embryo. So that tells you that the regulation of expression of that gene is going to be largely at the level of protein synthesis or translation. And sometimes you find cases like that, where the messenger RNA is all over the place, but the protein is only found in certain parts of the embryo. So it's a powerful technique, especially the two together. And you can look at embryos in different stages of development, looking for a specific messenger RNA. And maybe you find the messenger RNA is produced just right before the protein is being produced. But maybe not. Maybe you find in the egg itself, you find that messenger RNA in the egg or in a region of the egg. But you don't find the protein until much later in development. That tells you something. That tells you that that messenger RNA was laid down during oogenesis. It was stored in the egg, unused until the right part of development. And we do see many examples of that. In the lab, we mentioned that an egg contains proteins and messenger RNAs that are the program for early development in the embryo. So, these are powerful techniques, especially when you use the two of them together. Okay, now another thing you could do okay, don't worry, if you have any questions, don't hesitate to ask. All right, I will go to section number 2. Yes?
Okay, after you remove the chemicals, what's your next step?
Okay, well once you remove those chemicals, and once again, those chemicals separate out the two strands of DNA. They break the hydrogen bonds. Okay, so we separate. Now we already added the probe in before we removed the chemicals. So here's our labeled probe. And here's another copy of it. And here's another copy of it. We have lots and lots of probes. Now, when you remove the chemicals, hydrogen bonds between nucleic acids can reform. But it's much more likely, with all these probe molecules around, it's much more likely that the probe is going to stick to this strand of DNA first, long before this other strand has a chance to do so. So yeah, in other words, a probe vastly outnumbers the sister strand of DNA. So that's the idea. When you remove these chemical agents, hydrogen bonds can reform, but the probes much more likely stick to one strand of the target DNA than its sister strand. Did that help?
Yes, but then how are you seeing where it's localized?
Oh, the probe's got something, a label. Fluorescent dyes are common, radioactive isotopes, which expose film, and there's other kinds of detectors. Or sometimes even things like enzymes that leave a colored precipitate. Any of those ways will work. So, and the key thing is the probe has to be labeled because otherwise you simply wouldn't know what was there. Okay? All right, we're going to try